So I hope you had the time to play with multiple of the things that we've discussed last week. And what I'm going to be doing today is continuing, of course, as we started last week in our voyage through neural networks, and particularly biological neural networks and spiking neurons. So I wanted to start out with the outlook of the overall arch, like story arch, that we're having this year in primary neural networks. Here, we started lecture 1 with computational neuroscience versus neural computation, and we already started also introducing the Hodgkin Huxley model. You've done a project probably, and you've gone through on multiple aspects of that project. I am presuming that the project was relatively low level, I mean, straightforward. I'm hoping we're going to be looking into some specifics. I want to know how the message got across and whether you had fun. I didn't get many questions through Discord for that particular project. We will still be looking into questions for the projects throughout the duration, so you can continuously ask questions about any project at any time. Then lecture 2, this lecture. We had a little bit, I've reconsidered what our material for each day was going to be a little. I didn't want to overburden lecture 2, going from models of spiking neurons all the way to integrate and fire networks. So I've decided to stop at integrate and fire neurons. And then on lecture 3, we're going to be focusing on networks, both spiking networks and non-spiking networks, looking into structure and function relationships. Then on lecture 4, we're going to be looking into the relationships between plasticity and learning. And on lecture 5, we're going to focus on patterns and perception. So precisely how learning bootstraps certain cognitive abilities, such as pattern recognition. And for the final encounter, which is our test day, we're going to have the project presentations. Today, I have released the themes for the projects in Brightspace. So this is the grand story arch. And then particularly for today, we will start out with a review of our Hodgkin and Huxley model and how you've experienced it. I mean, whether the main components of that lecture got across, that project got across, and that's going to go through a Mentimeter poll in a second. After that, we're going to follow it up with an Hodgkin Huxley review, which is essential for the lecture today, just to bind the concepts with the project material and create a continuum to what you're going to be looking into today, which is spiking and models of spiking, and our understanding of what are the ideal conditions for spiking and what kinds of spiking neurons can there be as a function of their conductance interplays. So this already happens after the first intermission. So threshold shenanigans, is there really a threshold in the neuron? That's the question for the time after the intermission. And we're going to go into neurodynamics proper, which is the dynamical system study of spiking activity of biological neurons. Then we're going to have an intermission. Later, we're going to look into the reduction of dimensionality. What are the strategies we can have to reduce the dimensionality of the complex Hodgkin and Huxley neurons into more amenable to analysis neurons? The neurodynamics part we're going to have just before is basically giving you the theoretical material that allows you to think about reductions of dimensionality in the context of spiking neurons. I'm going to talk a little bit about parameter spaces, so everything that can happen under the sun. And then for the remainder, we're going to be looking into another project, which is going to treat directly neurodynamics. And then we're going to have questions and answers in Discord. Without further ado, I want to bring you to a Mentimeter probe. So here's the code that I would like you to use, 261556. And I wanted you to gauge your own understanding of these fundamentals here. So many of these you have already encountered in previous studies, also during nanobiology, and the project should have bolstered your understanding, like filled it up with more content so that you can bring this knowledge forth. So I would like to hear from you. How do you feel about these particular concepts in neural networks? So time constants, activation variables, currents, conductances, capacitance, reversal and resting potential, whether they make sense to you. So very murky, very clear. Then based on your feedback, we're going to be looking a little bit more in detail to each of them in turn. Maybe also it's a good time for you to actually ask a question now. You can use the chat, I think it's most conveniently for this. And you can also ask your question via microphone if you feel so bold. And then we can move forward. So we have already 15 participants. There's a total of 30 people inside this room today. A slightly smaller number. I guess that people also trust that the lectures will be online, but you lose the moment, no? So we want to do this live. So I am a little bit surprised at the fact that reversal and resting potential is one of the shakiest elements of all of this. I understand on the one hand. I mean, like, so usually if we had more time for this whole course, I would focus on a project 0 that discusses the origins of the reversal potential and the resting potential more closely. In fact, given this feedback that you already had, I think what I'm going to do is I'm going to make it available to you, project 0, so that you can, if you feel shaky about the reversal and the resting potential, which are absolutely fundamental concepts of, cell physiology, I think that it's really good to strengthen that knowledge. And so I'm going to make a little project available that usually we wouldn't deploy, but you can then work through it at your own leisure to buttress this understanding. Now, I also understand, activation variables and time constants, where they actually come from, it's not immediately clear. But on a grand average, you gauge them fairly high. So currents seem to be obvious. The activation variables times the conductances become the currents. So in fact, you do need to understand the activation variables to understand the currents that are coming in the cell, or at least the fluxes. And capacitance seems also to be fine. So basically the rearrangement of the ionic or charges in the cell. And the important thing that I wanted to bring up here for capacitances is the opposite relationship that it has with the leak. It looks similar and equivalent from certain respects, but we have to keep it in mind that the capacitance is acting on the change of membrane potential. So it's basically a dampening at the level of the membrane potential voltage change, whereas the conductances and the currents, the current-driven conductances, ionic conductances, they're coming in the cell, and they don't need, so they coming in the cell immediately, and the capacitance basically dampens the rate with which the conductance can change the voltage. So hope that this is going to get another up in the slides to follow. So thank you for this. I would like to continue with this quiz. So also, if you have specific questions, is a great time. So to ask in the chat. I'm going to move to the next one. I wanted to also gauge your feeling about, as you start working with Python, you've been thrown, I mean, if you haven't done Python before, you've been thrown in the deep end of the pool immediately. And, oh, that's nice. That's very reassuring when the first one kicks it on to 5, all the end of the spectrum. I can very well imagine that Brian, particularly, is a little mysterious in certain aspects. And this, is okay. But I wanted to share with you this notion of simulator language on top of a programming language, the simulator language. What is it doing in terms of solving these differential equations? So in fact, it's the forward Euler solver. I should actually put here also a question about, have you ever heard about forward Euler or forward method of solving differential equations? These days we're teaching them in computational science ever since this last year, but this is really what Brian is doing in the behind the curtains. It's solving the differential equations via an ODE 4-5 solver, for example, or a forward Euler solver. But this is great. So so basically you feel very secure about how to use Colab. You understand what initialization is doing. You're able to produce the answers by, either by shift enter the code, or by actually, so how I meant the question is in terms of the exercises that you've done. And what a neuron simulator is a little bit murkier. What I'm going to do, given this, and I'm going to note it down for myself, I'm going to run a 5-minute short snippet video explaining what the neural simulator does and how it ties in with Brian. I'm going to do this offline. I just wanted to have this feedback first, and so appreciate it. Now we have 22 responders out of 30 participants. We have 25 in the one before. So I think that that's okay. I will move to the next slide if you don't mind. And this is about the exercises themselves. So particularly part 2 of the project, I don't actually even know if you went all the way to part 2. I would be happy if you went until the end of part 1, just executing the code that was already there and trying to understand it. That's actually more than enough for the purposes of the course. But nevertheless, I wanted to know how compelled you felt to go through this. Okay, that's not too bad. I mean, like enjoying debugging is a tough one, isn't it? So I absolutely understand. So also for people that didn't know Python before, I imagine that producing new code would have been a challenge, and we were not so expected maybe copying parts. That would have been useful, I mean, to get a hang of it. But I imagine that this 1/3 of people that actually produced new code were the people who were a little bit more confident already with Python. For those of you, you haven't actually asked much in Discord, so it seems to be very self-explanatory. Brian, too, as a simulator, has many points of failure, and it's particularly difficult to debug because of the 2 levels. One is the programming level, and the other one is the consistency of the equations level. Units become very important to Brian, and particularly that the state variables are all there wrapped inside your neural group correctly. And so it gives messages that might be particularly difficult to debug if you weren't given the units ahead of time. If you design your own model, this is one of the possible pains that the Brian involves. On the other hand, by having this strict requirement that the units should be consistent in the system of units, international system of units, you get a very physically meaningful model. And this is incredibly valuable at that level. So you are dealing with state variables that very closely reflect what's going in the cell. And so it's understandable that you might struggle to get Brian's models working, and this maybe also relates to the project that we're going to be deploying today. But on the other hand, I wanted to give you the view that this is very useful to think about. I mean, to lock in those variables and units as particular representatives of this kind of modeling we do. So thank you very much for your feedback. So most of you read and understood the code, produced 1/3 produced the code. That's kind of like what I had in mind. It's always good. The code ran in the end. So 50%, that means that some people tried and failed. Come back to us. I mean, like, copy some error messages, dump them some screenshots with feedbacks from Brian, and we will be able to help you as well. If you want to do a little bit more Hodgkin Huxley problem today, you're also welcome to. Okay, so this is then 15 minutes for this part of the feedback. I would like to quickly open it again for questions. And I have to always bring the chat to this page I'm handling here. And I wanted to now review the Hodgkin Huxley, the most important concepts, maybe tying back to what your insecurities about it may have been, particularly activation variables, because those are very central to the understanding of Hodgkin Huxley's, not only, but conductance-based models. Hodgkin Huxley is the mothership of a great class of biophysically meaningful conductance-based models. So the executive message here is that the current going through the circuit is the sum of the individual ionic currents. And as you've, one of the things that I wanted to hone in here today in this equation is the number of dimensions, effectively, of this model. So when we speak about dimension of a dynamical systems model, and we're going to come back to that, we're really talking about the state variables. And the state variables are the ones that indicate what's the current state of the model. For example, the reversal potentials are not state variables. They're parameters. They're things that happen in the cell. They have to do with the concentrations, but we imagine them as being largely constant during the action potential, or during the activity. They can change, and there are actually models that make them change, but it's a solid simplification to imagine that they are actually fairly constant. So they're parameters, they're not variables. Then you have the maximum conductances, such as G bar, GK, GL, G bar K, G bar sodium, G bar leak. Those are maximum values representing the maximum number of ion channels. Those are also parameters. But if you want to know what your neuron is like right now, given those parameters, you need to specify, in the case of Hodgkin Huxley, 4 numbers. I should actually have asked this as a question in the group chat, but I think that you kind of figure this out. Think about it for a little moment. So which variables would you need to specify to actually know what is the state of the entire state of the neural network, of the neural model you have? So you can calculate the difference of voltage and membrane potential if you have knowledge about what's the proportion of sodium channels that are open, what's the proportion of potassium channels that are open, and what's the proportion of sodium channels that are inactivated. So N M, and H are three fundamental variables that indicate the state of your neuron. And V would be the master state variable, the one that requires all of the activation variables to basically be computed. So the four state variables here would be V, M, N, and H. The differential equations we have, V dot, N dot, M dot, and H dot, tell us how they change over time. So if you want to know the state in the future, what you need is the state of the moment plus the rule of how the state changes. We're going to be looking into this from the neurodynamics perspective in the second half of the class. But for the moment, save the number. Hodgkin Huxley has 4 state variables. The simplification of the equation, you might have wondered about the negative sign. There is a comment in the project about this, so go back to it. We're going to actually also go a little bit more into it. But in principle, the capacitive current, which is a function of the differential of the membrane potential, plus all of the conductances should be equal to the overall current coming through the circuit. And here we have the GI. This is actually a function for every single ion. So I here is the ion. So it could be sodium, could be potassium. It could be many other kinds of ions. And regulated by the particular activation mechanisms that we imagine in the ion channels. So here the sodium channel having both an opening and closing, M, activation variable that goes from 0 and 1, so proportion of channels open or close, and H, the actual inactivation variable. M has a exponent of 3. You said you understood most of you understood the activation variables, but this is something that actually comes up often as a doubt. Why the cube? So there are multiple reasons for that with different levels of analysis. One is, first and foremost, because Hodgkin and Huxley needed the cube in the equations to fit the action potential well. So that's number one reason, and it's basically a fitting reason. So when they fitted it, or the equations originally, they've discovered that if they had the cube on the activation variable for M, the onset of the action potential was faster in the model. So it was convenient to have a exponent there to make sure that the rise is fast, goes from 0 to 1 very quickly. Now this is the pragmatic reason, if you like, so the fitting related reason. But more interestingly, it turns out that this cube relates to the number of degrees of freedom of the sodium channel, of the squid in this case. So you have 4 subunits, and one subunit is the inactivating subunit. That one doesn't move, but the other 3 can change their conformations. And in order for the sodium channel to be letting sodium ions through, you need the 3 subunits to be in the open conformation. So basically, if you think about M, the activation variable, as being the probability of a single subunit, if you have the 3 subunits independently moving, probability would suggest that in order for you to have the channel open, you will have to have M times M times M, so M cubed. And similarly to the potassium channel, M to the power 4th. So that actually is a prediction of the Hodgkin-Huxley about what the structure of the ion channel is. And that's what's kind of mind-blowing, that after fitting this the channel very closely, Hodgkin and Huxley come up with the discovery, still to be done, like 40 years in the future, after crystallography of sodium channels, that this exponent relates to a particular structure. So this is obviously a very powerful model prediction, and that's, you know, one of the many reasons why they're so deserving of the Nobel Prize for physiology back in the day, because these predictions are embedded in the Hodgkin Huxley model. So the exponents here then relating to degrees of freedom generally, but also obtained via fitting often. We haven't talked much about the experiments that are used to measure these activation variables. So where does these cubes come from? And many of them come from experiments such as the ones that I've asked you to do in the project. For example, you can give a holding potential, giving a holding current, and see what happens at the membrane potential. With in the back of your mind, thinking that whatever happened in the membrane potential must have to do with the state of the activation variables. M is related to the sodium. M is related to the steady state of M, and the steady state of M is related to the membrane potential. So first you change the membrane potential, then the steady state moves to a different position. So for example, so if you give a very positive membrane potential to the cell with a current clamp, then very quickly the steady state changes immediately. It says, okay, we want to be open because we are in a very high membrane potential, and so sodium subunits all get their open conformation, making M close to 1 in the steady state. But until we get there, we still have to open the channels. So we know that the steady state at that membrane potential is a certain current. You obtain this by looking at the relationship between the current injected, the voltage that you have, and what is the new membrane potential. So after choosing a holding potential, you see how many currents, sodium currents, came in, like via the fast deflections here. For example, I don't know if you can see my cursor, I hope you can, but when you change the stepping potential in this voltage clamp, you have a fast and a slow component. The slow components relating to the steady state, and fast component relating to the transient. And so you make these experiments multiple times in the cell, and you gather values for your proportion of channels open, imagining that if you have a very high potential, you have maximally open, you have a very low potential, maximally closed. Things in between relate the current to the activation variable. Hope that was clear. So these are the experiments that one would do to, oh, thank you for the feedback, yes. So this is the experiment then that you would do to measure M. You can inspect this slide closely. It's also figure 2.10 in Izhikevich's book. But basically, you relate the activation to the voltage current relationship. So it's so-called the IV curve. So what you see here in the panel indicated by the cursor is the IV current. So you give a membrane voltage via a voltage clamp to the cell, and you measure the current that is necessary to keep that holding potential. But by that, you can then infer what is the proportion of channels open. And so you obtain your M activation variable that looks like a sigmoid from experiment. And that's why we use the sigmoid also to represent it. And so here we also have a tau. You may have noticed that the taus for sodium are incredibly fast. So the time that it takes for the sodium channels to open after a voltage deflection is very small. So if you see tau here is at level of 1.2 milliseconds. That's experimental data. You see actually the the error bars. The fact that the error bars are squashed here with very high voltage holdings is the fact that it's so fast changing the membrane, the changing of the membrane potential to the steady variable, that it's almost negligible, you see. And really so the model that I presented to you of the Hodgkin Huxley is not strictly the one that has been proposed by them. I prefer going for a model that has a little bit more inherent simplicity and generality about it. So I chose to present you with a model where the activation variables are sigmoids and the tau variables are Gaussians. And the reason for this should be somewhat intuitive. Not only it comes from experiment, but also in the case of the sigmoid, you go from all channels closed to all channels open, but you go smoothly, right? So as a function of V. Now the tau, there is a maximum duration that it takes for your channels to open and close, and that one is at the place where you are right in the middle of your sigmoidal curve. There's a relationship between the two. Actually, that's not a necessary condition, but for intuition, it's sufficient. It's important to then look at it. So to understand really the Hodgkin Huxley is to understand the activation variables. And here we have the steady states where we can use our mind's eye to imagine what would be the actual state of the Hodgkin Huxley momentarily if the voltage deflection was changed. So say here we are at where my cursor is pointing. So notice that these activation variables here are the shifted potentials from Hodgkin Huxley, minus 65 millivolts they've added to the resting membrane potential because it made their analysis easier. But you have to remember that it's actually the resting potential is these values minus 65 millivolts. Okay. Anyways, so if you're here at where my cursor is, you can see that you have some open channels for M. You have most of the channels deactivated. So you have a little bit of sodium current available coming in, making the cell depolarize. But you also have open potassium channels, a certain proportion. And they're balancing each other out. So this is what's called a window current. So sodium is constantly coming in, potassium is constantly going out. And so there is balance between these two currents because, have open potassium, open sodium, they're balancing each other out. In the back of your minds, you should also remember that there's the sodium potassium pump that is actually equalizing the concentrations in the cell. But if you're thinking about electrical currents exclusively, you're thinking about this so-called window current here. So there is a point in the Hodgkin Huxley where a certain current is constantly coming in and a certain current is constantly coming out. But V dot, that is dV dt, that is the change of membrane potential, is 0 because they're balanced. So this is the window current. I wanted to add this slide particularly so that when you're revising this, you can see what you can remind yourself that during these in this interval you have a window current. Now back into the generation mechanisms. This should make a lot of sense now, and I think that the majority of you already did, but I get back to it. Hope that you're able to understand all of these subcomponents. They are all equally explanatory. I'm not going to go into this because the project was about this. Hope that you've done the initial part 2 of the Hodgkin Huxley project. It's reproducing this figure, and so you can get even more intuition by looking into the interchanges between H, N, and M. Now, some of you may have noticed that in part 2 of Hodgkin Huxley project, in one sub-exercise, I have specified some values for M, N, and H, and V with many numbers after the comma. And you might have wondered, where do these numbers come from? So, a quick explanation. Brian starts the state variables, if you do not initialize them, with zeros. But if you initialize the model with zeros, the model has to relax until it's equilibrium. And so if you start, for example, with 0 sodium current coming in and 0 potassium coming out, and you start with a 0 membrane potential value, you actually have to go from 0 all the way to minus 70 millivolts, because that's where the resting membrane potential will be. And so that will create a transient. So you will see like a little bump that you might have noticed in some of the earlier simulations. And that bump is called a transient. It's the time for the time series to get to its equilibrium state. And in Brian, you can save that state, or you can observe that state and then use it later on, so as to have a start for your model that is already at a resting state, so that you don't need to waste that computational time for the transient. And so that's also why here you can see that the gating variables have initial values, and those are at the equilibrium. So the values that I have are the equilibrium values. One more way to look into this, the conductances for sodium. So the resting membrane potential is effectively a weighted average of the conductances and the reversal potentials, as you can see here, Vrest. Now, when you have your membrane potential here where the gray arrow is, you see that as a low, very low membrane potential value, the sodium current is very small. So the weight of it, there are very few sodium channels open. If you flick that membrane potential quickly to a positive value, the sodium conductance grows because the activation variable sodium opens, and your new resting membrane potential is very positive. So your cell, once you change your potential, will have a new powerful sodium conductance, at least during the action potential. This is just before the inactivation variables kick in. So you then have a very powerful conductance, which shifts the whole resting potential, and that's actually what pushes the membrane potential during the action potential to very depolarized positive values. Cool, right? So it's an interesting way to think about this. This is a fulcrum. So here the metaphor is that you have a lever, and the Vrest is basically a fulcrum, and you apply some weights on these fulcrum to set where the equilibrium would be. So I think it's a very powerful metaphor for the resting membrane potential during action potential. So here's rest, here's action potential. And ultimately, the action potential is made of these two positive and negative feedbacks. If you did not have inactivation variables of sodium, then once you inject a little bit of voltage, sodium channels open, and that makes the resting potential grow. Like so, our new membrane potential a little bit higher. You continue to add current via sodium channels that are opening. So more channels open, the more voltage, the more channels open. So that's the positive feedback here on the left. If you did not have the potassium or inactivation channels, then that would actually take the resting potential to this new value here, very elevated at 60 millivolts, close to the sodium reversal potential, and stay there. So the action potential will actually just be a toggle, which would go from low resting potential to a higher resting potential if there were only sodium channels. But since there are potassium channels that activate with a high voltage and inactivation channels that activate with a high voltage, you actually push the resting membrane potential again down. And that's what causes the refractory period and the hyperpolarization or repolarization of the neuron. Okay, so action potential then is a mixture of these two things in Hodgkin Huxley. We only have three currents: potassium, sodium, and leak. Maybe other current you were injecting. But I wanted to give you a sense of how many kinds of channels and their actions can be, and where they are located. So if you're thinking about a complex neuronal model, people have been measuring, the activation variables for different kinds of ion channels, and this is what they find. So we have this is on the x-axis. You have the half voltage of the sigmoid, basically where is the middle point of the activation. So you can see that, for example, potassium, our main potassium current here, IK, is activated at a high threshold and has a fairly slow time constant. We also have then the difference between the rectangles, which are the activation variables, and the inactivations of the currents, which are the ellipses. So this is just a sample of the kinds of ion channels that we can have inside the system. So the trends and currents then are the arrows. And so this wraps together what I wanted to tell you about the Hodgkin Huxley. So now we have 9:45. We started at 9. We're a little bit delayed, so I want to give you a 10-minute oversight of what compartmental models are before we go into the next step of our lecture. So the Hodgkin Huxley model is a bubble, and in that bubble you have ion channels around it, and this is a very good representation of a cylindrical squid axon. But obviously you know that neurons have dendritic trees, have axons. How do these currents relate to each other is a question. Had a little bit of an animated GIF at the end of the project to give you a view of how saltatory conductance works like, and now I want to give a couple more theoretical angles so that it can help think about these. And also I'm going to give you a couple of empirical results of using compartmental models instead of what are called single compartment models. So the Hodgkin Huxley original is a single compartment model. We can easily extend it into a multi-compartmental model. So in the compartmental model, we see the neuron as thousands of small cylinders. This cell is a Purkinje cell from the cerebellum reconstructed from anatomy. So people first trace the cell in the microscope, and then they measure the diameters, and they approximate the complex morphology of the cell, which kind of wobbles, with these cylinders. It's a very good approximation at first, and you can actually show this. By modeling the cell as many compartments, you introduce some boundary conditions. For example, the dendrite has a tip, and so at the tip, if the dendrite is very small, then there is no current coming out of it. Shape matters quite a bit to the activity of the cell. So a cell with the same densities of ion channels will actually act very differently. So reconstructions become a very powerful way to look into the class of what a certain type of cell can do. And another thing that the compartmental model allows you is to model the different compartments with different ion channel densities or distributions. Just let me focus on this last topic quickly. You have G bar for sodium, for example, and that represents, in the Hodgkin Huxley model as we had, millisiemens per centimeter square. Why centimeter doesn't matter, could be micrometer, of course, but anyways, it's a density. It's a number of channels per unit surface. So that applies generally for any kind of compartment. So you can, for example, have a constant maximal conductance for all compartments, because after all, they're all made of membrane, and the membrane has the same available number of spots for the channels. But if you know a little bit of biology, you know that things are never that simple. And so what actually tends to happen is that axons, somata, and dendrites might express different kinds of channels. Particularly in the soma, you have a lot of sodium channels, and even more so in the axon hillock, in the first initial segment of the axon. You have a lot of sodium ions, and that's where the action potential in most cells is generated, because that's the place where the maximum positive feedback takes place, lovingly. And in the dendrites, you have other channels. Maybe we have a current that is hyperpolarization activated. So compartmental models allow you to have a description of the cell that takes into account the differential expression of densities of ion channels along this morphology. And these interchanges are very fundamental to certain activities of cells. The cell, one of the cells that I study in the inferior olive, has very fundamental differences between soma and somata and dendrites, and I will show you a nice video of them next lecture. I'll come back to this point. But here are the things that might come completely unexpected to you and are results of compartmental modeling. For example, on A here, you have a compartmental model that has been given a steady current. And that's a single neuron that has been given a steady current multiple times, one after the other. And as you see, there is quite a bit of variability in the different traces. So every single spike train, like they're same cell, multiple trials, and so the trials are diverging. They produce the same spike more or less at the same time, but as time goes on, they start to drift apart. So there's a lot of variability in what the neuron is doing if you're giving him a constant, or if you're giving it a constant current. Surprising fact appears when you actually inject. So you could say, well, neurons are not reliable. They are biological things, and then they do every time different things, and the variability is enormous, so there's a lot of entropy. In comes B, our panel B. And in panel B, we also have multiple trials, only that now it seems like it's only a single trial because they're so perfectly aligned. What we've done now is we've injected current that is a noisy current, frozen noise as we call it. So the same fluctuating current every time with the same charge as being delivered by this step in A. And you see now that that noisy current creates spikes at the right time. Most of the trials, there's a couple trials that do something a little bit different here, but for the most trials, you have almost exactly the same spike times. Very reliable. This is a result from 1995, and it has been reproduced by a compartmental model of the pyramidal cell very closely. This actually, in the Hodgkin Huxley, is harder to reproduce. Another interesting consequence by the same group, Mainen and Sejnowski, they had like one science and one nature paper 1 year after the other. simple modeling work. They're just, you know, how nice it is to be at the crisp of the zeitgeist wave. So what they showed here is that a neuron with the exact same sodium, potassium densities in the different parts of the cell, these are different cells, A, B, C, and D, all of them have the identical densities for the conductances. So the value for G bar for every single compartment is identical. And so by changing the cell morphology, these are reconstructed cells, you can see that they are doing something completely different. So the first cell is irregularly firing, the second cell is adapting in the lower rate, the third cell chatters because it produces a bunch of spikes and then it silences, and even more so in D, which is a pyramidal cell in the human cortex, or cat cortex, I think in this case maybe. And so important to think about how morphology might influence, even though you have the same parameters for all of these cells, same parameters. The only thing that is varying is the morphology. You have this fundamental difference. How do you get to that? Well, the trick here is that you're basically cutting the cell in the cylinders, as I mentioned before. And these little compartments that represent the dendrites in a certain way, they have, so each of these cylinder pieces, here, for example, in B, this guy, sends current to the guy above and sends current to the guy below and receives from their neighbors too. And this is how we model it. We model it via these axial resistances here. Do you see RA? So RA is the component that is coming from the neighboring compartments. You can also model this as a second order differential equation, but this is already discretized in space. I don't think it's worthwhile to go into the actual derivation of the second order differential equations. Much easier to think about already discretized. And so here in the bottom equation, what you get is on top of your usual dynamics from the currents, here the sum of currents in K, these are the different ion currents. You have the contributions of the neighboring compartments. So in R prime A and RA, so these are the axial resistances, and V prime M minus VM. VM is us, and V prime is the guy to the right. I don't know if I have actually one of the videos in front of you. So V prime is to the right, and V double prime is to the left. And so these are the contributions that are coming from a radial axial. It's going along the axial dimension with the resistance, and that resistance is a known parameter. So in principle, we are actually just simply assuming that there's conservation of charge. What comes in, what comes out. So if you have here now a discretized axon into many compartments J, and you have then time in T, if you start an action potential at the first compartment, J1, that propagates via charge to all of the connected compartments without loss. At least at the radial dimension. It could very well be that you do get leak across the outward membrane, but not within the neuron. And so you see here on the left panel, you see the 50th compartment and the 70th compartment, and you see that it took about 20 milliseconds for them to propagate from one to the other. This is just a demonstration of conservation of charge in compartmental modeling, which is the basic assumption underlying what we just saw in the previous slide. And the differences in compartmental modeling I had already mentioned, but here is an interesting one in terms of synaptic integration. So this is a granular cell of the cerebellum, and here we talk about synapses. These granular cells receiving an inhibitory and excitatory currents from synapses in the same compartment or in different dendritic compartments. And we plot the activity at the somata of the cell. And here what we see is that if you excite and inhibit at the same time, but in different dendrites, in this particular cell model, you actually produce a spike, but you do not if inhibition and excitation coincide in the same dendrite. So this gives yet another free dimension for the, or another degree of freedom, a complex degree of freedom for the synaptic interactions at the level of dendritic trees. Okay, so flyby, I showed you this in the first, now you have much more material to comprehend what was done in the reconstruction and simulation of neocortical microcircuitry. We've done a lot of compartmental models that were based on reconstructions with differential expression of different conductances in axons and dendrites, blue and red, here in different layers of the cortex, both for excitatory and inhibitory cells. So these are the cells from the somatosensory cortex of the mouse that have been reconstructed to emulate the activity. And this picture shows then on the left the cells in this column that have been reconstructed. In B is a spike raster for each for the different cells in the different layers. In C, you can see the action potentials themselves with some chattering neurons, some regularly firing neurons. And on D, you see probability of spike counts. We're going to be looking a little bit more into spike counts of networks in our next lecture as well.